Hi everyone. Today we are going to continue with the chapter 3, and we have discussed the plasma membrane of the cell. So chapter 3 discussed about the cells, the living units, and we discussed already the plasma membrane, and we are going to continue with the rest of the cellular structures. So let's start talking from the cytoplasm. All cellular material which is located between the plasma membrane and the nucleus is considered as the cytoplasm, and it's composed of cytosol, inclusions, and organelles. The cytoplasmic organelles, based on whether it does have a membrane or not, it can divide it into two categories, either membranous organelles or non-membranous organelles. So mitochondria, endoplasmic reticulum, Golgi, peroxisome, lysosomes comes under membranous category, including the nucleus. The non-membranous would be the ribosomes, cytoskeletal, and the centrioles. The membranes, the importance of membranous organelles would be that the membrane-bound organelles allowed compartmentalization of the units inside of the cell, and that will improve the proper function of the cell. Let's talk about different organelles now onwards. The mitochondria is called as the power plant because it produces the energy currency ATP through the aerobic respiration, and the aerobic respiration is the respiration which requires oxygen. And it's a double membrane organelle, and it's inner membrane. The inside of the mitochondrial membrane is being folded into cristae, and this cristae is involved with the cellular respiration, and that also increases the surface area of the inner membrane of the mitochondria. The other important fact would be, other than the nucleus, mitochondria is the only organelle which contains DNA. In addition, it does have RNA and ribosomes. With related to the ribosomes, it's a non-membranous organelle. It's the site of protein synthesis. It's made up of protein and ribosomal RNA, which is rRNA. And there are two switchable forms you can find in the cell, which are the free ribosomes, which is floating in the cytoplasm, cytosol, or you can find ribosomes bound to organelles, which are the membranous bound form. And endoplasmic reticulum is one of the membranous, one of the organelles where the ribosomes bind, and it does have a particular functions too. So endoplasmic reticulum, it consists of series of parallel interconnected cisterns, and the cisterns are flattened membranous tubes that enclose fluid-filled interiors. So if you see here, these are the cisterns, and they're very flattened, and inside you do see that the fluid-filled component. And these teeny tiny dots that you see in yellow are the ribosomes attached to these membranes. The other important aspect would be the ER is continuous with the outer nuclear membrane. So this is the nuclear membrane which is depicted in purple, and of its outer side, you do see the endoplasmic reticulum is attached to it. Based on whether the ribosomes are attached to the ER or not, we can divide the ER into two types: rough ER, where the ribosomes are attached to it; the smooth ER, where the ribosomes are not attached to it. And the rough ER, it is the site of synthesis of proteins that will be secreted from the cell, and it's the site of synthesis for many plasma membrane proteins and the phospholipids. When the proteins enter into the cisterns of the ER, they get modified as they wind through the fluid-filled tubes, and the final protein is then enclosed in vesicles, and they are sent into the Golgi apparatus for the further processing. With related to the smooth ER, they are kind of a network of loop tubules continuous with the rough ER, and there are enzymes found in its plasma membrane, and we call them as integral proteins, and they function in lipid metabolism, which is lots of cholesterol and steroid based hormone synthesis happening at the smooth ER. They make lipids for lipoproteins. They involved with the absorption, synthesis, and transport of fats, detoxification of certain chemicals, converting of glycogen to free glucose, and also storage of release of calcium. So in that way, whenever any cell does this function more, you would expect it to see more smooth ER in these cells, especially like with related to cardiac muscles, heart muscles. When you require lots of calcium, you do see lots of smooth ER in these cells. So the next connection with the ER would be the Golgi apparatus. It's a stacked and flattened membrane cistern sac. So it involves with the modifying and concentrating and packaging proteins and lipids, which is received from rough ER. When I highlight in red, it indicates that it's important. So if there's a question like where does the modification and packaging of proteins and lipids happening, then you have to know that it's in the Golgi apparatus. In this process, 3 steps are involved. So what are those? The transport vesicles from the ER, they fuse with the cis or the inner face of the Golgi, and then the proteins and lipids taken inside and further modified and tagged, sorted, and packaged. And they have going to be trafficked down, and it's been controlled. So that's why it's called a traffic director. And the final product is now going to put out or pinch off from the strands or the outer face of the Golgi. So let's look at what we have just discussed in this figure. So you see the transport vesicles coming from the ER, which is the endoplasmic reticulum. And this is the inner surface, the cis surface of the Golgi, and they get combined, fused with the inner surface of the Golgi apparatus. The proteins transported through its cisterns, and it gets modified during its transportation, and they pinched off from its outer surface, which is the trans surface, and the these vesicles would be maybe fused into the plasma membrane, and then like in this figure, it can put out from the cells. So once again, the cis surface where it receives from the vesicles from the ER, the trans surface is the one which gets pinched off, and then vesicles which leads to destination. The other membranous structure organelle you find in the cell would be the peroxisomes. So it's a membranous sacs which contain powerful detoxifying substances, and it neutralizes toxins. And when cells undergo lots of metabolic processes, it produces lots of free radicals, which are toxic and highly reactive molecules. And they're, of course, natural byproducts of the cellular metabolism. So 2 enzymes involved with this process in the peroxisome, they are the oxidases, which uses oxygen to convert these toxins into hydrogen peroxide. And hydrogen peroxide itself is toxic to the cell as well. Now, then it has to be further neutralized. So then the catalase jumped into the role, which it converts the H2O2 into, which is the hydrogen peroxide, into harmless water. So oxidase acts first. It converts the toxic into the hydrogen peroxide, and then the catalase jumps, and then it converts into the water. And peroxisomes, they play a major role in the synthesis of fatty acids. So where there is a fatty acid synthesis production cells involved with, you would encounter lots of peroxisomes. The lysosomes, even though it sounds like peroxisomes, it's another spherical membranous brown structure, but it contains digestive enzymes, which is acid hydrolases. So what they do is they digest ingested bacteria, viruses, and toxics, and they degrade non-functional organelles. You would find lots of lysosomes in macrophages, like lots of immune involvement cells, which acts as a first line defense in our immune system. The metabolic functions would be they break down and release glycogen, break down and release calcium ions from the bone. And if they release its content into the cell itself, it will autolyse the whole cell. So it digests themselves, and the process is called the autolysis. So why we learn these things is you will encounter when you practice, there are some diseases which involve with these organs. For instance, the lysosomal storage disease, it will result when one more lysosomal digestive enzymes are mutated and the lysosomes do not function properly. And there's another condition called Tay-Sachs disease, where the lysosomal enzyme needed to be break down glycolipids in the brain cells. They are not functioning properly, or they lack it, and then these patients accumulate lipids in their cells, and that would affect the brain function, and these patients may result with seizures or mental retardation, blindness, and mostly they do die before the age of 5. So considering all the membrane system, including the ER, Golgi, secretory vesicle, lysosomes, as well as the nucleus and the plasma membrane, we all together called it as the endomembrane system. And these membranes and organelles work together in order to produce, degrade, store, and export biological molecules, and to degrade potentially harmful substances. Other than this membranous structure, the organelles and the non-membranous structure, the very important part of the cell would be the cytoskeleton. And it's an elaborate network of rods that run throughout the cytosol. It acts as the skeleton of the cell itself, and it's composed of major 3 types of filaments and tubules. They are the microfilaments, intermediate filaments, and the microtubules. So they're in order of the increment of their size. The microfilaments smallest, intermediate in between, the microtubules are the largest. So microfilaments, they're the thinnest of all cytoskeletal elements. The protein involved is the actin, and each cell has a unique arrangement of strands, although they share a common terminal structure. And some are involved with cell motility, changes in cell shape, endocytosis process, as well as the exocytosis. And here you would see the 3 structures, which is, you see this is thin, this is large, the microtubules, and these are the intermediate ones. So of course it's in actin subunits, you would see it's a twisted form. And here you have tetramers, like it's like 4 ropes twisted together. It's much more give a toughness for these intermediate filaments. And here you would see that it's kind of a little hollow structure, but still it's composed of subunits. The intermediate ones, as I mentioned before, is size between the microfilaments and the microtubules. They're tough, insoluble, rope-like protein fibers, and they're composed of tetramer fibers, which is twisted together, resulting in one strong fiber, and that would help the cells to resist pulling sources. And some have special names, such as like neurofilaments in nerve cells, keratin filaments in epithelial cells, as such. On the other hand, microtubules, they're the largest of cytoskeletal elements, and they consist of hollow tubes composed of protein subunits called tubulin. So easy to remember, microtubule, T-tubule, tubulin, and which are constantly being assembled and disassembled, which is like they're forming and disforming, forming and disforming. It's a constant process happening. And Microtubule, they most radiate from the centrosome area of the cell, which is like the cell center. Microtubules involved with determining overall shape of the cell and the distribution of organelles. Many organelles, they're attached to microtubules to keep organelles in place, and many substances, they moved throughout the cell by motor proteins, which use microtubules as their tracks. And the other important aspect would be the centrosome and centriole. So these are two different things you have to remember. So centrosome, which is located near the nucleus, means as the cell center. And centrioles are the pair of barrel-shaped microtubular organelles which lies right angle to each other. And these centrioles form the basis of the cilia and the flagella. So you see these are the centrioles and they have two. They formed in right angles to each other. With related to this structure, skeletal structure, we would encounter some cellular extensions. So certain cells have structures extending from their cell surface. You might have heard word cilia, because in the respiratory tract you get it's ciliated epithelium. So cilia and flagella aid in the movement of cell or materials across the surface of the cell. And there are something called microvilli on the cells. Some cells, they are the finger-like projections which extends from the surface of the cell to increase the surface area. Cilia, furthermore, it's a whip-like motile extension of the surfaces of the certain cells. As I mentioned before, you would encounter it in the respiratory cells. The flagella, on the other hand, they're long extensions and they propel the whole cell. So mostly you would see it in sperm's tail, which propel the cell. Both structures, of course, cilia and flagella, they're made up of microtubules and synthesized by centrioles that are called the basal bodies because they form the base of each of the cilium and the flagella. So it's important. And they do have two structural differences. Cilia and flagella, of course, they have 9+2 pattern, which is like microtubules of 9 sets is there. At the same time, they are in doubles. And you will see when you see the picture what I meant about this. And it's the double tube surrounding a central pair of the doublets. And slightly different from 9O pattern of the centriole is the 9 triplets with no tubules in the center. So in addition to this pattern, it does have the cilia movements, makes an alternate, the movement of the cilia happens with the power strokes, which is happening alternatively, which is the power stroke and the recovery stroke. So this alteration produces a current at the cell surface, and that would leads to movement of the substances. So here you would see like 9 sets of 2 microtubules, and there is 2 centered microtubules in the middle of the cilium in here. And here you would see a basal body which has a triplet. Actually 9 outer doublets of a cilium extend to the base, and these triplets are joined together. So there's no centered 2 tubules in here. But if you here, you would see again the 9-2 pattern. This shows the power stroke and the recovery stroke. The power stroke propels, it's a propulsive stroke, and the recovery stroke is the returning stroke, which brings back the cilia back into its original position. With related to the microvilli, they're finger-like extensions of the plasma membrane, and you would encounter them in very absorptive areas such as the intestine, the kidney tubules. It increases the surface area, and of course, they're made from actin filaments. The other important, very important organelle would be the nucleus. It's the largest organelle which contains the genetic library of an individual, and it's involved with synthesis of nearly all cellular proteins. And most cells, they're uninucleate, which is like they have only one nucleus, but some do have more than one. Example would be the muscle cells, skeletal muscle cells, and certain bone cells, some liver cells, and some do not have nucleus at all. So those are the red blood cells. The nucleus, we can divide them into three major structures: the nuclear envelope, nucleoli, and the chromatin component it does have. So nuclear envelope, it's a double membrane barrier which encloses the jelly-like fluid, the nucleoplasm. So nuclear envelope is a double membrane organ. There are two organelles which is in the cell double membrane, the mitochondria and the nucleus. And it does have some pores which allow substance to go in and out, but still it's going to be guarded by nuclear pore complex. And that nuclear pore complex regulates its substances. The nucleoli is the other component of the nucleus, which is a dark staining spherical bodies within the nucleus, and it's involved with the ribosomal RNA, which is the rRNA synthesis and assembly of the ribosomal subunits, because ribosome is composed of two subunits, the large subunits and the smaller subunit. And also nucleoli is associated with the nuclear organizer regions, which contains the DNA and that codes for the rRNA. Usually one or two nucleoli exist per cell. The chromatin is another important component of the nucleus, which is it consists of combination of DNA, histone proteins, and RNA. So 30% is from DNA, 60% from histone proteins, and 10% is from RNA. And they're arranged in fundamental units called nucleosomes, which consist of DNA wrapped in around histones. And chromosomes, they are condensed chromatin. So remember, chromatin is composition of three things. And this condensed state of those chromosomes helps to protect prevent breaking the chromatin threads during the cell division process. So in this figure, you would see that this is circular structure would be the histone protein, and this purple ribbon-like structure would be the DNA. And the DNA is wound around this histone protein, and lots of histone proteins wound together and then combined and condensed together and form this chromosome structure. So it's like you take a bead, you take a ribbon, you wind the ribbon around the bead, and then another bead wound around the ribbon wound around the bead like that. You have several thousands of beads, and then you further wound those beads together and then compact those things, and that's how you make the chromosome structure. So imagine when you loosen it up, it will take much more yards, even though it compacted into a smaller area. Okay, so this is the end of the presentation too, and we will see you from the next presentation, and you will see that in my other recording. Thank you.